Had a fantastic morning. Welcome to the live lesson. I just realized again that my camera is blurry again. So let me just quickly wipe that. Need to make sure I clean it before we do this again. There we go. Okay. Welcome everybody. I hope that paper one went really well for everybody. And that it wasn't too bad. I thought that paper one wasn't too difficult, to be honest. And just so you know, what does that mean? So what does it mean if paper one is easy? It can mean two things. One, it can mean that paper two is going to be more challenging, which generally it is. I need to prepare you for that. There's going to be a lot more like interpretation and thinking questions in paper two. What it also means is when paper one is so easy, the memo is very strict, which means that you have to give what they are looking for exactly as they ask. And sometimes that is the downfall for a lot of pupils. They think they've given the right answer, or they think they've given what they wanted, and it hasn't turned out that way. So I'm just preparing you for that. But with that being said, I thought the paper was easy enough that the answers were very straightforward. There were some weird questions. I thought the weird one about the auxins and nature conservation was out of nowhere. I thought the one about why Africa has less severe head injury records. You know, it's not weird. It's just so unusual for them to have those kind of like suggest questions. There was also a sneaky question in the A, B, both or none, which I'm actually not going to talk about right now, but I myself am questioning the very last one, 1.3.3. Some people say it's both. When I talk to my other teachers, they say it was none. I'll elaborate that a little bit. I'm thinking about doing a memo video for you guys. So let me know if you want to see a memo video, and I can give you what I think the memo would have been for the exam, so you can check what your answers were. So I think what I'm going to do is I'm going to start, and I'm going to begin with our suggested questions that came from the comment section of the other members. And I'm going to start here with a pedigree diagram. And I chose this one because it links well with what I feel like might come up in the paper on Monday, which is a sex-linked disorder. Now, remember, everybody, a sex-linked disorder, it can be any sex-linked disorder. It can be colorblindness. So I specifically chose this one because maybe colorblindness will come up on Monday, maybe not. But I think that sex-linked disorders is a popular one that they were going to choose. So let's go through it. Okay. It says color blindness is a sex-linked, and they actually tell you that disorder caused by a recessive allele. And these are important pieces of information. It's also important to note, grade 12s, they don't always tell you it's recessive. Sometimes what they'll do is they'll say it's sex-linked, and then they just give you this letter over here with the little lowercase d. And the lowercase d is the important part. That's the bit that tells us this is a recessive disorder. If this was a capital D, then it's a dominant disorder, okay? All right, then it says the diagram below shows the inheritance of the disorder in a family. And if we have a look here, let's just take a little bit of time to fill this out. Now, I know this sounds very taxing and it's a lot of labor to do, but I always tell my grade 12s to go and actually fill in the individuals, because at some point they all play an important role, particularly the people who have been named, right? They're probably the ones that are going to be involved in a question. So the first thing I want to do with the people who have names, I want to zoom in on them, is I'm going to give them their sex alleles first, okay? So all the circles are females, and if they are empty like this, they do not have the disorder, whereas if they are colored in, they do. So what I'm going to do is I'm going to put in their sex alleles first. So those are all my females. And then I'm going to do the male as well. And he's XY. I am going to do the dad as well, even though he doesn't have a name. And you'll see now why I like to do the parents, because the parents reveal a lot about the children, right? So now what I want to do is, looking at the key, fill in the letter D, depending on whether or not they are colorblind, right? So Thuli is not colorblind. Now, the only way that she's not colorblind is if at least one of her X's is a capital D. But I'm not going to fill in the next one just yet. Okay, I'm going to leave that because her children will tell me what her other X is. If we go over to the dad, we see that he is colorblind, which means his X carries a lowercase d. Remember, nothing on the Y. This is an X-linked sex disorder. Then we go down to the children, and the children will also help us finish up what Thuli is. If we go to Molly, she doesn't have the disorder, so that means she has at least one capital D. Senzo, on the other hand, has the disorder, and he has inherited a lowercase d. Now, I want to point something out. Senzo is really important in this family tree because, remember, boys inherit their Y from their dad and their X from their mom, which means that this D must have come from Thuli, which then allows us to complete the picture for the mom. She's lowercase d. Then they have a daughter called Linda. Now, Linda also reveals potentially what Thuli was as well, because she gets one X from mom and one X from dad. And so that means she got a lowercase d from dad and a lowercase d from mom. Now we go back over to Molly just to make sure that we have all her pieces. If she got this capital D from mom, her other X must come from her dad, which means she is a carrier of the disease and she gets a lowercase d. Okay, so we've filled in the important people in this family tree. We might need to go back and fill in some others, but let's get into the questions. So first things first, give the phenotype of Senzo. Now, I want to just caution you grade 12s. They like to do this. They like to go pheno and genotype back to back. And they do that in case you don't realize that you've changed from a pheno to a genotype answer. So just be very wary of that. Don't give two phenotypes, one after another, if you know what I mean. Be very, conscious during your exam. So, give the phenotype of Senzo. So we just go up to our block over here. We look at what Senzo is. So he is a colorblind male, and that is his phenotype. And if you have any questions during the lesson, don't forget to put in the question section, and I'll have a look at it now when we go through it. Then it says the genotype of Thuli. Well, we've already solved Thuli earlier on. So she was a X with a capital D on it and a lowercase d on it. So she's a carrier. Now, I did see some questions pop up on the live chat, so I'm just going to have a look at them. So the first question is from Muhammad. Could you say that the mother is homozygous, non-color blinded? I think like two capital. No, so the mom can't be. I see what you're trying to say. The mom cannot be homozygous. She cannot be homozygous, everybody. Why? Because she has a colorblind son, and sons only get their Ys from their dad. That X cannot be passed on to Senzo. One of these X's must go to Senzo. So the only way Senzo is colorblind is because the mother carries the gene for that. And that's also why sex-linked disorders are so closely associated with females. It's because most females are carriers, but they do not actually experience the disease themselves necessarily. Wonderful Wise asks, why is Linda's genotype not X lowercase d, X capital D? Okay, so Linda is not this, by the way. That's what you're saying. She cannot be that because she is colorblind. So she has to have the disorder. And the only way girls can have the disorder is by having two lowercase letters. So she cannot be this. That's why. Okay. All right, let's move on to our next question. So it says, describe how Linda inherited colorblindness. So I want you to pay attention as to how I answer this question at grade 12. This is very important. Whenever they talk about how an individual inherited any kind of disorder, this is what you are going to always do. Okay, I'm going to go onto a blank page to show you. You must structure your answer as follows. You must tell me what mom has and therefore what she gave. Okay, so that means, who are we explaining? We're explaining Linda. So mom was X, capital D, X, lowercase d. She gave Linda an X with a lowercase d. The second part of your answer must be, what is dad? So what does he have? And then what dad gave, right? So we go over to dad. There's dad. We solved him. I knew we were going to need him for one of these questions. So dad is X small d y, and dad gave X small d. The third and final part of your answer, and normally these questions are out of 3, is you are going to summarize this and say the following. Okay? Because Linda is XD, she will be color blind. And that is how you are going to structure every single one of your how to explain inheritance. So if they ever ask you, how did this person inherit their disorder? How did this person get it from their parents? Something like that. You're going to tell me what mom is, tell me what dad is, what they gave, okay, and how when those two things come together, they produced a certain product, which is this. And that is how you're going to get 3 out of 3. It's sometimes out of 4, but mostly it's out of 3. Like this question we've done now is out of 3. This one here, describe. It's actually out of 2 even, so it's even less. You probably still get the marks. Okay, I saw a question, so I'm going to quickly look at it. Chelsea says, can females have and present with hemophilia? Very good question. About a couple of years ago, there was a hemophilia question, so also prepare for that, everybody. In the final exam on Monday, they can also ask you hemophilia. So this female over here is an example of a female who is a carrier of hemophilia. You can also have this, which is a female who doesn't have hemophilia. And then you can also get this. Now, this is a female who has hemophilia, but let me explain something. Females with this genotype often are not born or do not survive birth because hemophilia affects your blood, and because you have a double copy of the broken or mutated allele, you don't survive. However, and this is a big however, in the final exam, if you get a girl like this in the family tree, okay, it is not an error. That's not what there's no error in the paper. What they are trying to do is say, hypothetically, like pretend that this is possible. Pretend that we can have a hemophiliac female and solve for the problem, solve what we're asking. So, can you get a female who's a hemophiliac? Technically no, because they cannot be born. However, if they are given to you, like it's in the family tree, for example, it's in the diagram like this one, like they've colored her in and they've said she has hemophilia, work with what they have given you. It's not an error, and answer the question like hypothetically she could be born. Okay, all right. Let's move on to our next question. It says, explain why generally more males than females are colorblind in a population. So it's an explain question. Remember, what is my technique? We need a statement and some reasons. And this one is out of 4. So we are going to definitely need 3 reasons, right? So our statement is going to be as follows. Colorblind or colorblindness is on the X chromosome, right? And our reasoning is linked to now the recessiveness and how you inherit it. So colorblindness is on the X. It is recessive, okay? Reasoning, therefore, a male has 1X will have colorblindness. Next reason. Females have 2 X chromosomes. Therefore, it must be present on both. And you can sort of summarize this last little bit, say, so we see it more commonly in males. So essentially, to answer that kind of question, you have to follow my structure of give the statement, color blindness is on the X chromosome and it's recessive. Therefore, if a male has one X with it on, he will have color blindness. However, females have two chromosomes, therefore must be present on both. So we see it more commonly in males. Okay. I'm just going to quickly look at your questions because I can see there are a couple of them. Mohammed asks, is hemophilia always caused by recessive allele? Yes. Nyoko, how do you answer a question when they say, explain why a female who has hemophilia will not survive? If they ask you if a female is hemophiliac and why won't she survive, that is because hemophilia is a blood clotting disorder. And if your blood cannot clot, it also means that your blood can be quite, let's say, unstable inside of you. So why can't females be born hemophiliac? They cannot be born as a hemophiliac because their blood composition will not allow for their organs to develop, and their blood will not circulate nutrients and oxygen. So the actual blood cells do not form properly, and you end up not being able to give birth to that child. So 2 reasons: the blood composition is not correct, therefore your organs cannot grow properly, and your blood doesn't clot. And number 3, the blood struggles to circulate nutrients and the fetus or the embryo dies. That's basically why you cannot have a hemophiliac female. Okay, right, let's move on to the last question. Molly married a normal male. Use a genetic cross to show the possible geno and phenotypes of their children for 6 marks. Now, let me say this, grade 12s, this is 6 marks for free. Why do I say that? Because you all should be able to do a genetic cross, right? If you can't, I'm about to show you how to do it. Let's just make sure I've got all the people. So we need Molly and a normal man. Let's go up here. So Molly is heterozygous for her 2-letter D. So I'll just remember that she's a carrier. And grade 12s, I want you to pay close attention to how I set out my answer. I'd like to point out I do mark paper 2, which means I have like way more insight or experience into paper 2. And I'm going to tell you exactly how I mark it. So if this is how it comes across my desk, I'll give you full marks. So first things first, we're going to write P1 in the margin, right? That means the parental generation. Then we are going to write what Molly and her unknown partner is. So we're going to say female. Oh, actually, I'm not going to say female. We are going to say non-color blind female. Right? Why non-colorblind? Because she is non-colorblind. Below that, we are going to write her alleles. And I feel like I have now written this really large that I'm not going to be able to fit in the man on the side here. So I'm sorry to be irritating, but I'm going to actually zoom in and write it closer. So we have non-color blind. Yes, you must write female, by the way. You must always identify if it's male or female. And then below that, capital D, lowercase d. Okay. Then we're going to cross her over here with a male, and he is also a non-color blind male. You could say normal if you want. That's also okay, by the way. And we're going to go X, capital D, Y. Okay? So now I zoom out. So now you can see everybody there. There they are. Okay. Now what we're going to do is we need to make their gametes. So we're going to write the word meiosis. Okay. And then below that, we are going to write in the column over here, gametes. And now I'm going to take these individuals, and what I like to do is I like to put circles around them. And let me tell you why I like to put circles around the gametes. As a marker of the final paper, too many matrics write these too close together when they do their gametes, and then I mark it wrong. By putting the circles around it, you navigate around that issue. So what I do is I do this, and then I put a circle around it. And I put the other allele in its gamete, and I do the same for the males up here. Circle. It actually just represents they are in, so it would look something like that when you're done. Okay, then just below here you're going to write fertilization. Yes, the placement of the words is important, like that. Okay, then in the margin we're going to write F1, which means filial. It means children. And I use a Punnett square. I know some of you might use a line diagram, but that's really up to you and what you've been taught in school. But we do line diagrams at my school. And like that, right? And I'm going to put in my, not line diagrams, we do Punnett squares. Please make sure you don't split the female. So what that means is if you're going to put her letters at the top, you must put both of them at the top. For the male, you must also keep his letters in the same column. And now we multiply into the Punnett square, right? So we've got X capital D, X capital D, X capital D, X lowercase d. Then we have X capital D, Y, and X lowercase d, Y. If I went too quickly, I do have a monohybrid video to explain how I did that, but you're just basically multiplying these letters into the sort of Punnett square. All right, so now let me show you the part that people get wrong, most common or commonly, which is the pheno and genotype writing. So let's start off with the genotype writing. Okay, so how many of these individuals have the same genotype? So we're going to have to put them into males and females. So right now, looking at all of them, they are all actually different in genotype because this female's genotype is different from this female's genotype. This male's genotype is different from this male. So we've got 25%, and I'm going to just zoom in a bit here. We've got 25% X capital D, X capital D. We have 25% X capital D, X lower, 25% X capital D, Y, and 25% X lowercase d, Y. So none of them can be grouped together is the important bit I'm getting at here. Okay? Now, let's do the phenotype because this is the one we get wrong. So I want you to pay attention to this. When it comes to sex-linked disorders, this is why we get it wrong. If you look back up here at our genotypes, I don't have to show you the Punnett square. Which one of these females, or both of these females, exhibit color blindness or no color blindness? Well, this female is not colorblind, and neither is this female. I repeat, you do not write the word carrier ever. I'm going to say it again in case nobody heard me, but you never write the word carrier. There's no such thing in a Punnett square genetic cross like this. If you can talk about a carrier in an explanation, but you cannot use the word carrier in this cross, okay? So when we group these two ladies together, 50% female, you could say non-colorblind, you can say not affected, you can say whatever, you can say normal, whatever, as long as it's saying that they don't have the disorder. For the males, on the other hand, these are two very different ones. So we've got 25% male, also non-colorblind. And then we've got 25% male who is colorblind, right? So I want to reiterate that. You do not use the word carrier. If we go back up to the top here, did you see she is a big letter, small letter? She's technically a carrier, but did you see I wrote it anywhere? No. If you look down at the bottom here, under genotype and phenotype, under phenotype, I grouped the two females together. Why? Because they do have the same phenotype. Neither of them are colorblind. You do not need to separate the carriers. The carriers must be grouped with the normal or unaffected females. Okay, I'm going to have a quick little look at your questions in the chat. Let me just open it. Okay, let's go. So, Wonderful Wise says, ma'am, how do you identify generations in a pedigree diagram? So that's super easy. I can just show you. The generations are these dividers here. So this is generation 1, this is generation 2, and so on and so on. So each one of these lines is a generation. So that's how you would determine it. Right, then next question from Marina. Could we say non-affected female or male? Yes, you can. Wonderful Wise, can you put circles around the blood groups too? Yes, you can. Muhammad wants to know, will they ask a dihybrid cross? If they will, could you please? Of course, we're going to do one. It's actually one of the next few questions that we're going to do, definitely. Okay, everybody. Prepare yourselves for one of these. There will be something like this on Monday. And I think that the only other thing I want to say to you before I go on is if they ask you how many females or males of the group, please make sure that the question says of the females or of the males. If it just says of the children, it means everybody. So to make this simple, I'm just going to add in a page to just show you what I mean. This is important. That's why I want to show it to you. If the question says, how many of the males have the disorder? So let's just pretend here's a rough family tree. And this is another family branching off here. Okay. So let's say this male has it, this male has it, and let's say 1, 2, 3, 4, 5. Yeah, okay. So this is the, let's say this is the pedigree diagram I'm going to give you. And the question is, how many males? They want to know how many males of the family have it. So what that means is of the males, not of all the family members. So I will say that again. If they say how many of the males have the disorder in the family, what that means is they don't mean the whole family, right? So we've got 1, 2, 3, 4, 5, 6, 7, 8 people in this family. They are not asking all the family members. They're saying how many of the males. Now, how many males are there? 1, 2, 3, 4, 5. So out of the 5 males, 2 out of the 5. Not 2 out of the, how many did I say we have? 1, 2, 3, 4, 5, 6, 7, 8. Not 2 out of the 8. That means 2 out of the 8 family members, including the females. But the question asks of the males. So how many of the males? 2 out of the 5 males. I just wanted to clear that language up because sometimes that's what trips us up in the exam. Okay. All right. We're going to change gears now and go into DNA. We are going to do a dihybrid question. Don't worry. It's on the cards for us to do. So let's go on to the next question. So number 2 says the sequence of amino acids in a protein molecule is coded by DNA and RNA, and the table shows some mRNA codons. Oh, I don't want to cross that out. So it shows some mRNA codons and the corresponding amino acids. So we've got our mRNA codons and our amino acids. So number 1, according to the table, how many codons code for phenylalanine? So we've got phenylalanine over here, and we've got phenylalanine over there. So it should be a fairly straightforward answer in that it is 2, right? Moving on. What is the anticodon for glutamic acid? So we go up to the paragraph here and we find glutamic acid, which is GAG. That is the codon. What is the anticodon? So we'll be on the other side, like on the tRNA. It would be CUC. So that's going to be our answer. CUC. Easy peasy for now. Right. Moving on. A section of mRNA has the following base sequence and is read from left to right. There it is. Give the DNA base triplet for the last codon of this mRNA section. So we go to our section here. It says the last codon, which is that guy over there. Remember, they want the DNA base triplet. So they've given us the codons, the mRNA codons. That would mean if I want the DNA bases, it would be T, G, G. Okay. And yes, I'm allowed to write T because it's asking for the DNA base triplets. Okay. And this is mRNA. Right. Moving on to our next question. Give the first amino acid coded for by this section of mRNA. So we're looking for GAU. So we go up to the table and we look for GAU, which is aspartate. Just to make sure there's no other GAUs here. Yep, aspartate. So that's going to be our answer. Nice and easy as well. Okay, just like that. Right, next question. Gets a little bit harder now. A mutation occurred which resulted in the following base sequence on the mRNA molecule. Now, what you need to do is spot the difference between these two. So which one has mutated? GAU, same. CUC, same. GAC, same. GAGC, there it is. So there is our mutation. The rest are the same. And just check, okay? Don't stop. Keep making sure they are all the same. So this is the one that has changed. That's the mutation that I've highlighted. Describe the mutation that has occurred. So what we have here is the letter U, right, has been substituted in for the letter C. And that is what you are going to describe. You are going to describe what substitution is, which is also known as a point mutation. That's what you might have learned in class. And you are going to then say that C has been replaced by U. Okay, right. Now it says, explain the effect of the mutation described in question 214 on the resulting protein. Now I want to say this, grade 12s. Please make sure, and this is why I chose it, because it lines up with my prediction and what sometimes they like to do to you and to try and catch you. Please make sure that if it did mutate, it actually makes a different protein. And it's not just the same protein, okay? Sometimes it's the same protein. I want to catch you out. So let's actually just make sure. Let's originally look at what AGC was. Let's change the color. AGC is serine. It then became AGU. Let's have a look at the table. AGU is also serine. Do you see how they're trying to catch you? If you didn't check, what would your answer been? You would have said, oh, well, it's mutated, so it's a different protein, it's a different amino acid, it's a different this, it's a different codon, it's a different no, it's not. It's not. They tried to catch you out here to make sure that you actually did the work properly and to figure it out, but it will stay the same. And to also sort of give a clue to how short your question is, or your answer is, it's only for 2 marks, okay? So that then means for this one here where it says explain, again, you can make your statement and your reason, but your statement is going to be simply the mutation has not changed the resulting protein. Reason, because even though the codons are different, the amino acid is the same. And that's all you're going to write. I'll say it again. Statement, the protein will be the same. Why? Because even though the codons have changed, the amino acid is the same. And that's it for 2 marks. Okay. Last question on this one before I look at our questions in our chat. Name and describe the process occurring in the nucleus which results in the formation of mRNA. And that's for 6 marks. That, everybody, is going to be the process that we make mRNA is what? It is transcription. I'm not going to write out the whole process of transcription because I don't think I need to. But the best explanation for that comes from your guideline, hey? That's the one that you must write. I know you might have learned a different one in class, but trust me, write the one that's in your guideline, not the one that your teacher has given you. I even tell my students, don't write the one we do, write the one that the guideline gives, okay? I'm going to look back into the chat now for anybody's questions. Okay. Sangita, how was blood group O formed? I'm going to get there soon because I'm going to do some blood groups later, promise. Unati, if they ask about children, do we look at the 2nd generation going down? Yes, you do. 100%, you're correct. Mohammed, how does mRNA have adenine in it? Doesn't RNA always have uracil? Mohammed, no. To answer your question, just so that you know, DNA has the following. It has T, A, G, and C. RNA has A, U, G, and C. So the only letter that's replaced between the two is the T and the U. And that's because, if you know this, these two are grouped together and these two are grouped together, like these two and these two. Okay. Hope that clears that up for you. And last question, wonderful wise ma'am, how do you tell the difference between translation and transcription in diagrams? Lovely question. I can quickly draw you how to tell the difference between the two. Okay, so this is transcription. Okay, a couple of things to look out for in transcription in the diagram. Number 1, there will be a nuclear membrane. Okay, that's the first thing. That's the first clue that it's transcription. There will be a nuclear membrane. Number 2, the mRNA is still inside the nuclear membrane. This is mRNA. Another thing is, what have I drawn here? That's not what I want to draw. I want to draw something like that. Bear with me. I'm just drawing a section of DNA. If this is the section of DNA in there that we want to copy, let me draw it better actually, everybody, because I feel like I'm letting you down on that drawing. Let me do it properly. Let me do it this way because it's easier for me. Okay. So this section over here is the section that has unraveled. It's the part that we are going to copy. That is the next clue. If there is a piece of DNA in the picture and a nuclear membrane and a piece of one-sided mRNA, in other words, a single strand like this, single strand, you know you are doing transcription. So that's the first sets of clues, right? This is the most important one though. If you can see the nuclear membrane or you're inside the nucleus, this is transcription. If you can see the mRNA being formed, it's transcription. If you can see the DNA that has unraveled partially, and this is the section of DNA that we're copying in here, we're in transcription. Translation, how to know that you're doing translation. This is also a nice, easy way to look at it. You're going to have a ribosome. That's the first clue. If you can see a ribosome, you are in translation. If you can see a tRNA, which sort of looks like this, you are in translation. If you can see a group of amino acids joining together, you are in translation. Okay. And last but not least, there is a piece of mRNA. This is mRNA inside the ribosome. And those are all the things that you look out for in translation. So first of all, ribosome, tRNA, is there amino acids present joining together, or is there mRNA inside the ribosome? That's how you tell the difference. Okay, I hope I've answered all your questions. I'm going to move on to our next question, which is going to be the dihybrid question, right? So there will be one of these probably tomorrow somewhere, but no, you'll never have to solve a full one, which means you won't have to do like a Punnett square with 16 squares, hey? don't don't worry, you won't ever have to do that. You don't have time to do that anyway. That's why they don't do it. All right. Mendel observed some characteristics of the pea plant, which he suggested were controlled by inherited factors. He conducted a series of experiments in which he crossed pea plants with contrasting phenotypes, which means they're like opposite, to obtain the offspring of the F1 generation. At first, his crosses were simple and involved only one pair of characteristics. Mendel counted the number of offspring showing each of the variations, and here is his results. So I'm going to take a few seconds to just break down his results. So I want you to notice that we have part of the plant. So we've got seed, pod, flowers, and stem. The characteristics, though, with the seed was like seed texture, seed color, pod texture, pod color, flower color, location of flower on stem, height of stem, right? And then he tells you all of the crosses of the P1s. Was it a round seed, cross a wrinkled seed? Is it a green pod, cross a yellow pod? A violet flower, cross a white flower? And then what's really great is in this column, which is important to us, it tells us all of the outcomes, and it tells us what all of the dominant characteristics are, because that was the point of Mendel's experiments, to prove that dominant and recessive alleles exist. So like there's dominancy, which means when you cross a round and a wrinkled, round is dominant. When you cross a yellow and a green, yellow is dominant. Is it a full pod or constricted? It's a full pod that's dominant. So all of these are the dominant characteristics. Okay, we call it a test cross. So these are all the tests that we did. And it is when they are what we call purebred crossing as well. So it's purebred round cross purebred wrinkled. What do I mean by purebred? For example, if round is the dominant, it would be like a cross like this. It would be R times little r. That's what I mean by purebred. It means the one parent is homozygous dominant and the other one is homozygous recessive. They're like pure. They're not mixed like a heterozygous, right? Okay, so we've interpreted this. Now we need to go down to the questions. So number 1 says, give the term for the inherited factors that Mendel was referring to. Those inherited factors, so the factor, everybody, is a allele. That's what he's talking about, okay? For B, it says a cross involving only one characteristic. That is a monohybrid, right? A dihybrid, remember, would be 2. Then it says, name the female structure of the flower where meiosis occurs. Now, don't be thrown by this. Don't be like, whoa, is this grade 11 work? Like, when did we learn this? Yes, they are allowed to ask this because you are supposed to have knowledge from all your years. Remember that, hey? You can't just rely on your grade 12 knowledge. I know that sucks, but they expect you to have knowledge from grade 10 and 11. And just think about it logically. The female structure in the flower, it's actually the same as what's in a human female, right? So you should actually know this. It's the ovary of the flower. Not too bad, hey? Just like in a female and a human female, it's the ovary. And I think that's why they asked that one, because it's not different from a human. Right, next one. Use the information in the table above to give the number of each of the following. Okay, so the number of characteristics of pods, and they're also going to ask for the alleles for the seeds. So let's start off with how many characteristics of pod do we have? So if we go back up here, how many characteristics? You go under the characteristic tab. We've got texture and color. So the answer there is 2. But now I want you to pay very special attention here. How many alleles are there for seeds? So now this is, and it says seed characteristics. So do you know what a lot of grade 12s do? They just go back up here and they go seed characteristic, 2. No. These are the phenotype characteristics. They want to know how many alleles. So you've got to go over to this column. There are 1, 2, 3, 4 alleles, because there's a round allele, a wrinkled, a yellow, and a green allele. So don't be trapped by the question. There are 4 alleles for these seed colors. It then says, for our next question, give the characteristic that is dominant for the flower color. Well, we just need to take that from our paragraph, our table up here. So our dominant flower color is violet, right? And then the next question says, who is the recessive for stem height? Well, if we go to stem height and we see that they all came out tall, the recessive must be short. Okay, so that's one way they can ask a dihybrid. There is another one that I'm going to include and do later on, where I think you're more, you're asking for that kind of question, where it's like, fill in the missing information, like what's in the diagram. So I think I'm going to do that now, because I'm going to add in that question, if I can just find it in my pictures at the top here, and then we can do it together. Just give me a second, and while I look for it, you can ask any questions that you might have. Where is it? Oh. Let's see, it's somewhere here. Is it this one? No, it's not that one. I'm gonna look for it now. Is it this one? No. It's one of these ones. No. It's not with a table. Let's see here. Maybe it's a newer one. I put it in my video for you guys to look at. I can't actually see it now. Give me a second now. I'm not disappearing. I'm just going to go into my pictures now and see if I can find it. So I'm still here, everybody. Don't worry. Where is it? Oh, there it is. After... There it is. This was the one I'm going to do with you guys. I think this is what you want to see as well for a dihybrid, right? This kind of thing. So let's quickly do this one. This one's nice and easy. It says a certain plant species has the following alleles for the characteristics. So we've got number of seed pods, big P, little p. So one seed, 3 seeds, and leaf shape, normal shape, wrinkled, right? So those are the dominants with the capitals and the recessives with the lowercase. The table below shows the results of the offspring produced by the genetic cross. So this is our table of results. And I just want to point out one thing to you. You should interpret this table before you start answering the questions. So do you see that these numbers actually have a pattern to them? And I know you're looking at it and be like, man, what pattern are you looking at? Well, I just want you to keep in mind that dihybrids have a ratio of 1 is to 3 is to 3 is to 9, right? That's your ratio that you should know. And that follows this same ratio, okay? And it sort of reveals what is and is not dominant. What we need to do is we need to solve for this table here. So first things first, we've got how many genes of the plant are considered here. So what is the difference between a gene and an allele, grade 12? So I just want you to think about it for a second. The difference between a gene is a characteristic. An allele is a version of the characteristic. So think of it like this. You get your flower color, that's the gene. Flower color, that's the gene. But the allele is the purple allele, the white allele. So those are variations of the color. So how many genes of the plant are considered here? Number of seed pods and leaf. So there's actually only 2. Don't count these. These are the alleles. These are the genes. So there's actually only 2 genes that we're doing here. Then it says, name the dominant phenotype of the plant. So the dominant, remember, is the capital letters. So the dominant phenotype is going to be one seed, because that's the capital P, and then for the capital L, it's going to be normal shape leaf. Okay. Then for number 3, it says give the genotype of each parent. I love this because you're probably thinking, okay, how do I work out the parent? Okay, don't panic. Okay, remember that ratio I spoke about earlier? The only way to get that ratio, and it's a law, by the way, it's not like sometimes it is, sometimes it isn't. This is a law. The only way to get that 3:3:9 ratio that I just spoke about earlier is if both parents are heterozygous, which means there must be this. So that's the genotype of each parent. You can write it twice if you want, you know, like you can go times a PLLP like that, but it's not necessary. I know it's for 2 marks, but I'm telling you now, it's a double awarded mark, which means that this will get like a double tick. Okay. Last question for this dihybrid, number of offspring that are homozygous recessive for both the characteristics. So now what you need to do is you can go up to your table here, and they need to be homozygous recessive for both. So that means they need to be 3-seeded and wrinkled. So let's see here. We've got 3-seeded, yeah, and wrinkled. So we've got 32. And that's it. Do you see how easy the dihybrid is going to be on Monday? It's not going to be that challenging, I promise, everybody. I promise. Okay. Do we have any questions before I move on to our next topic? No questions. Great. Okay, I think what I want to do now is I'm going to move on to this guy over here, which is a natural selection question. Again, something that was requested. I also... Sorry, Wonderful Wise, you want me to explain 153 again? This one here. I think you want me to explain A. Okay, so I didn't even have to work out the parents because of the following. When you do a dihybrid cross of parents who are heterozygous like this, which means they have a capital and a lowercase of each of the characteristics, you always get the following ratio. 1:3:3:9. This ratio is very important. We use it for dihybrids. Now, what is this, ma'am? You've written this, what is it? It basically means you will have 9 children that will have a certain characteristic. 3 children will have a different. 3 other children will have another group, and 1 will be on their own. This, by the way, is not the only ratio you are supposed to know, everybody. The other ratios, I will point them out just now when we're done here, but they are also really important for explaining yourself. So how did I get this? Okay. The only way to get these children in this cross here, based off of these numbers, okay, and I can slot these numbers in. It's 32 is to 96 is to 100 is to 290. That is the ratio. It's not perfect, by the way. Yes, I know this one's 96. I know this one is 100, but it's never perfect in nature. But this is how we try and get it as close as possible. This is the ratio. So the only way to get this ratio ever, grade 12s, is if both parents are heterozygous. And that is why my answer for A is a heterozygous parent. P, big P, little p, big L, little l. That is why that is something that you have to know. You have to learn. It's not something that I just sort of, how can I say? It's not like an application question. It's actually just a fact. And you should be able to recall it. So I suggest you go back and look in your textbook for dihybrids and this particular ratio. And this ratio helps you answer this question. So this number represents the offspring ratio. For every 9, you have 3 of this group, 3 of this group, and 1 of these. And this is what you have here. Okay, if I factorize this down, in other words, if I simplified it, I would get something very similar to 1:3:3:9. It wouldn't be perfect, but it would be very, very, very close to this ratio. Okay, so that's how I got this one over here. Now, all the other ratios, by the way, that you are supposed to know, I'm just going to put them out there, besides this one, is, I'm just going to leave it like this. So that's the one ratio you need to know. You also need to know that if you cross 2 big letters with 2 small letters, it's going to be 100%. So there's no ratio for that one. If we're going to do big letter times big P, little p, right? We do a Punnett square of that quickly for my own benefit. Big P, big P, big P, little p. Big P, little p, little p, little p. So that's 3. So this is a 3-to-1 ratio. Sorry, I just wanted to do the calculation on that so I know I got it right. So when you cross 2 heterozygous parents, it's a 3:1 ratio. And I think there is actually a ratio for this kind as well. Let's just put it in here. Little p's, big P, little p, big P, little p. Yeah, this is the 50-50. This is the 1-to-1 ratio. So let me write that one out. 1:1. So do you see all these crosses with these parents, right? When you have a purebred cross, they're always going to be 100% heterozygous, which means this, right? There is no ratio for this one. However, if you cross 2 heterozygous individuals, there will always be a 3:1 ratio. If you cross a heterozygous with a homozygous recessive, it will be 1:1. If you cross 2 heterozygous parents in dihybrid, it will always be 1:3:3:9. So these are the ratios. These guys, these ratios here, these are the important ones to know. Okay, right. I think somebody asked a question about me doing meiosis, and we are going to do a meiosis question. Don't worry. It's coming up. Tristan, hi Miss Angler. In Gauteng prelim exam, they asked what would the effect of the mutation on the DNA replication be? Will you please explain how you formulate that answer? I can do that, but I'm going now back in time, Tristan, because I did actually answer this earlier on. So if you've only joined now, maybe you haven't seen it. Unati, how do you know the parents are heterozygous? Okay, again, I only know they are heterozygous because these numbers told me they were. These numbers, they told me they were heterozygous. Because when you take these numbers, grade 12s, and you simplify them, they give you a ratio of 1:3:3:9. So 32, 96, 100, 290. Those are the numbers from the table. If I take my calculator now, which I don't have my calculator now with me on my desk, and I don't have a calculator on my tablet. I don't think I do. Maybe I do. But if I simplify these, it will become 1:3:3:9. How do I simplify it? Well, I go, this is very sort of basic maths. You probably do this more in Mathslit than you would do it in regular maths. So you go 32 divided by 32 gives me 1. 96 divided by 32 will give you roughly 3 point something. 100 divided by 3 again will give you 3 point something. And 290 divided by 32 will also give you 9 point something or 8 point something. But it gets you this ratio. The only way to have these numbers in the children, the only way, is if both parents are heterozygous, which means big letter, small letter, big letter, small letter. How did I know that? What's the theory behind that? You need to go back and look in the section of your notes or your textbook on the dihybrid section, and it speaks about this ratio. It speaks about if you cross a heterozygous parent with another heterozygous parent. So if we cross these two, you will get this outcome. You will get 9 children with this physical appearance. You'll get 3 children with a different physical appearance. You'll get another 3 with a different physical appearance, and you'll get 1 with the final version of the appearance. So that ratio is something that you must learn. Okay, I have to move on, everybody, otherwise I'm not going to get through. Because I still want to do this natural selection question and this meiosis question before we finish. So this one shouldn't take us too long. It says there are 2 variations of color in color of king snakes. Some have a bright, colorful pattern. Others have a dull pattern. King snakes are non-poisonous to their predators. Coral snakes, I'm just going to change color now because talking about different snake, have a bright color pattern. They are poisonous to their predators, and it is their defense mechanism. Some scientists observed that where king snakes shared habitats with coral snakes, there were more king snakes that had bright, colorful patterns, and the diagram sort of shows this below. So just to unpack this, we've got 2 groups of snake. One that is not poisonous, but brightly colored. Another that is poisonous and is brightly colored. And the fact that these 2 have similar coloring is effectively what is assisting them in their survival. So this is what the population looks like. We've got coral, bright, and dull. And as you can see, the dull snake is not very common, but the brighter king snake is. So for 3, 2, 1, explain how the bright color pattern of coral snakes influences their survival for 3 marks. So there's our little explanation question as usual, which is what? Remember to give the statement, then the reason. All right. So, and in this case, we'll have 2 reasons, by the way, because it's out of 3. Explain how the bright color of the coral snakes, so the poisonous ones, influences their survival. So our statement. Bright equals poison. That's your statement, right? You can say the bright color indicates they are poisonous, which means what? Predators avoid them, right? Which then increases survival. Boom, done. That's all you have to write. So you have to say, statement, the bright color in the snakes means they are poisonous, which means the predators avoid them, which increases their survival. Or you could say the predators are less likely to eat the coral snakes or catch the coral snakes, and they survive. And that's all you need to say for the 3 marks, okay? The next one is where it gets a little bit trickier, and that's why I chose this question. It says, use Darwin's theory of evolution through natural selection to explain why there are more brightly colored king snakes in this habitat. Okay, so remember natural selection, you're going to take your answer from your guideline, right? They've given you one, they give you a template answer. They tell you in bullet point what to say. And you're going to talk about the bright king snake and the dull king snake with natural selection. So when you formulate your answer, and I'm not going to write out the whole thing to save time, but when you formulate your answer, please, grade 12s, when the guideline says favorable characteristic, please don't just write some snakes are favorable and some are not. No, you must use what the paragraph gave you. Please. So the favorable color, please write that. So favorable is the brightly colored, right? The unfavorable is the dull. You must actually write that. So when they give you an example that you must apply, make sure you mention all of the animals. So you'll say, there is variation in the population. There are favorable colored snakes that are brightly colored, and the unfavorable color is the dull snakes. Then you go on to say that the brightly colored ones, they survive, they reproduce, right? Whereas the dull don't survive, therefore they don't reproduce, right? Then you go on to say after the don't survive, don't reproduce, is that there will be more brightly colored alleles present in the population and fewer dull. And you must actually mention there are fewer dull because it's asking you to explain why there are more brightly colored king snakes. They are more brightly colored because the dull get eaten. They do not survive. It is less frequent. And you end off your answer by saying there is more alleles in the habitat with the brightly colored, there are more brightly colored king snakes in the habitat and they survive, and there are more of them. The proportion increases, something like that. Okay, it is for 6 marks, so you're going to have to say 6 things. There is actually 6 things sort of already hidden in my answer, because that would be 1, 2, 3, 4, 5, and then the 6th thing would be there are more brightly colored who are produced and less dull that don't, and therefore there's a higher proportion of bright, and that would get you your 6th mark. So please get that from your exam guideline and just insert the color of the snakes in your answer. Okay. Right, last question. Is this my last question? I do have another question. Potentially you might be able to get to that, but I'm going to do the meiosis one for you now quickly too. We must expect to see this. I chose this one also because it's a little tricky. The diagram below represents the distribution of the chromosomes in pair 21 as it appears in the gametes at the end of meiosis 2 in a human male. Okay, this is at the end of meiosis 2 in a human male. So, number 1, you will notice here that there is 2 cells with double chromosomes and then 2 with none, which means something has happened incorrectly, right? Something is not right in this picture. And hopefully your first thought is non-disjunction, right? They failed to separate. So explain why the gametes represented in C and D do not have any chromosomes. That's for 3 marks. What are you going to explain there? Statement, reason, right? Statement is the fact that non-disjunction occurred, right? Reasoning, your chromosomes pulled to the same pole, which means the opposite poles. Like, you know, they normally pull across to opposite poles. It means this time all the chromosomes pulled to the same pole, leaving no chromosomes to the opposite pole. In other words, if you look at our picture up here, they all got pulled into these 2 and nothing got pulled into these 2 gametes. So in actual fact, these should actually be in here. There should be 1 chromosome in each. Okay. Number 2, if gamete A is involved in fertilization, describe how this may result in Down syndrome. So if we took this individual over here and they technically then have 1 extra chromosome, what you need to now do is you're going to, as always, this is an inheritance question, by the way, just like we did with mom and dad earlier. So gamete A has 1 extra chromosome. So when you describe how this works, and you'll see this is a describe question now for 3 marks. Let me just show you what you would write. You were going to say something like this. We're going to say, gamete A has 24 chromosomes, which it does. I know there's not 24 in here, but they're saying, let's pretend that this was a full chromosome and now it has an extra chromosome in it. So gamete A has 24 chromosomes. If it fuses with a normal gamete with 23, it will give you 47 chromosomes, which is therefore Down syndrome. So how do you structure your answer here? Okay, just like you tell me, like, what's mom, what's dad? What is dad? What is mom? And when they come together, what is their product. So that's always how you do these inheritance questions, or you explain how do you get the child, just as we did at the beginning of the video. Tell me what dad is, tell me what mom is, and then what is the product. So that's exactly how you structure that answer. Okay, almost done. All right, due to the process of crossing over, the chromosomes in the diagrams A and B appear to be different to each other, which, as you can see, they're not the same. Identify the phase of meiosis during which crossing over occurs. That's a straightforward answer. That's going to be prophase 1, because that's the only time it takes place. It can't be in prophase 2, everybody. You should know that. Number B, describe the events during crossing over. So that's when you say things like they move via synapsis to form a bivalent. They touch at chiasmata, and then, I can't fit it in here now, but they exchange alleles. And that gets you 3 out of 3. That is the events of crossing over. So homologous pairs move via synapsis to form a bivalent. They touch at chiasmata, and they exchange alleles at those points. And that's it. Last but not least, explain the significance of crossing over in natural selection for 3 marks. Let me just rub out some of this so that we have space. So what's the importance of crossing over? The importance of crossing over is, and remember this is a statement, reason, situation. So our statement is we want variation. Reasoning, variation increases survival, and it increases the favorable trait. And that's your 3 marks. So explain the significance of crossing over. It introduces variation, which increases the survival of the organism by increasing the amount of favorable traits there are in a population. And that's it. 3 out of 3. And you should expect to see these kind of crossover questions as well. Singite said, is interphase in meiosis or mitosis? Interphase is in both meiosis and mitosis. Interphase is the in-between phase, everybody. It's the phase that the cell is just doing its like everyday job, right? Okay, last but certainly not least, everybody, I'm going to do this very last question over here. I'm not going to do both of them. I'm going to do this one. This is actually the one I wanted to do. It's the cloning question. Again, we might see a cloning question on Monday, or like either a reproductive cloning like this, or a molecular cloning question. We'll have to wait and see. And I just want to answer somebody. Marina, could we say 2n plus 1 instead of 47? Yes, you can. Yes, you can. So if you are trying to, she's asking about this question here. Do you have to say 47? No, you could also say 2n plus 1. Okay. All right, let's do this last cloning question. There's like a whole long paragraph here about Futi, the cow, who was the very first cow to ever be cloned in South Africa. So let's just quickly look at it. So the first cloned cow in Africa is Futi, was born in the Northwest, blah, 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 blah. No fertilization was involved in the production of Futi. She was produced from a single cell taken from the ear of the donor cow. The donor cow had a high average milk yield of 78 liters a day. Cloning allows for the production of organisms with desired characteristics. Remember, there's a difference between desired and favorable. Favorable is nature choosing. Desired is humans choosing. Some people argue that cloning reduces genetic variation in offspring with no further genetic improvement. Cloning is an expensive procedure and may not be economical for commercial agriculture. Okay, they've given you that information for a reason. They must have, right? So let's have a look. 231. According to the extract, state one advantage and disadvantage of cloning. So one advantage of the cloning comes up here, where we can make a high average milk yield, right? So we can have a high yield. According to the paragraph, oh, I've spelled yield wrong there. According to the paragraph, we can have a high yield of anything, right? So a high yield that we took from the paragraph up here. You could also say that we have a organism with a desired characteristic. That can be an advantage of cloning. A disadvantage, as we take it from the paragraph, is it's expensive, or you could also say that it is not economical. But I would rather go with the expensive one because that's just like way more obvious. Less likely to get it wrong. 232. State why the donor cell was taken from LMGC, which is the donor, and not any other cow. Well, we chose this lovely cow because she was able to have a high yielding of milk. So that's why we wanted to clone her. We wanted to have more cows that make the same amount of milk. So why did we choose her? Because she has a high milk yield. And it's only 1 mark, so that's why it's just a statement. Next one. State why an ear cell was used and not an ovum. And this is for 2 marks. So why did we use the ear and not the ovum? Well, we use the ear because it is diploid and has all the characteristics, all the desired characteristics. Oh, I don't think I spelled characteristics right there. Characteristics. Whereas the ovum is not diploid, is haploid and doesn't have all the characteristics. So that's where you can get your 2 marks. We're saying the ear is diploid, tick, because it has all the desired characteristics, tick. You don't have to go on and say the ovum is haploid and doesn't have enough, because you can see there's only 2 marks here. But for safety's sake, right, I would say ovum is haploid and doesn't have all the characteristics that we want. Okay, Sunita, your question. Let me just have a look at it before I go on. On the advantages of cloning, can I say no to fertilization was involved? No, that's not an advantage. Okay, there's not going to be an advantage that you need to give. The advantages are really only that it's she makes high yield or desired characteristics. Okay. Okay, next one. Briefly describe the cloning process for 4 marks. Now, I wonder if everyone can do this. I'm not sure if everybody can do this, but I have a video on cloning that I suggest you go and watch. But essentially, they have 2 cells. This one is going to be the ovum. This one is going to be the somatic cell, the cell they take from somewhere in the animal. This one has the DNA we want. This one has the DNA we don't want. So what do? We take the ovum and we cut out the nucleus. In the somatic cell, we cut out the nucleus. We just want the nucleus. We then put the 2 together. We plus these 2 together. So you end up with this. So we put the nucleus inside the empty ovum. We give it a little electric shock. And then we take this cell, once it starts undergoing mitosis, we put this cell back into our donor. So it's going to go into the donor of ovum. So this is now going to get put into this individual. Whoever gave us this ovum, we can put it back in there. So what are the words and the way you need to describe it? So essentially you need to remember it's a description. So you say how, when, why, and where it happens. So we take an ovum cell and we remove the nucleus. We take a somatic cell and we remove the nucleus. We place the desirable nucleus in the gutted cell. This is called a gutted cell, or the gutted ovum, right? We place it inside. We give it an electrical shock. It undergoes mitosis and is then placed back into the uterus of the donor of the ovum. And that's how you explain cloning. All right, everybody. And that's pretty much it. I think I've covered all the major questions that might come up in the final on Monday. Is there any other questions you guys want to ask me before I go? I'll give you a couple seconds to write anything in the chat. Also, while I wait, if there's anything that you are thinking of typing up now, don't forget to go and watch some of my videos on the different exam questions. There is questions for you for just members, but there is also a whole bunch that I've done for everybody. So go and watch and find them in the different playlists. There's a playlist called Tricky Question. There is a playlist which is just called Exam Questions. And then there's also the members only one as well, but that is just for members. But what I do is I walk you through various questions, and I'm hoping that you're watching them, because sometimes, yes, it's very easy to practice a past paper question on your own, but sometimes if you don't get to the same answer, you don't understand why, those are really, really useful. Okay. Sunita, you want me to do blood groups? I didn't, but I can quickly do it now. I think what you were asking was the various blood groups. I'll quickly throw it in now. So we've got blood group O, B, A, AB, right? Those are all the blood groups that you can have. And the alleles that go with these are only ever written like this. O is 2 small i's. B can be a capital like this with a little I, or 2 big B's like this. The same for A blood group can be this as well. And then AB is just A, B. Okay, so that's how our alleles work for our blood groups. And O is recessive to A and B and AB. So in other words, these dominate O. But A and B are co-dominant. So there's actually 2 kinds of inheritance happening here, if you didn't know that. For O, we experience complete dominance. So this is complete dominance. But for A and B, we experience co-dominance. Oh, oops. Co-dominance. And we know that they're co-dominant because they form A and B. Okay. Is there a specific idea around it, Sunita, you want me to go over? All right, ma'am, do we need to know every type of fossil? If you mean every type of fossil, actually, we never did a human fossil question today, did we? To answer your question, no, you don't need to know every single one. You need to know the names, Australopithecus, Ardipithecus, and Homo sapien. But if you're asking the fossil names like Mrs. Ples, Lucy, Latoli footprints, yes, you do need to know those names. You do need to know who Mrs. Ples is, who Lucy was, who discovered Lucy, where did they find Lucy, who found Lucy, all of those things. And as a member, you should be using my study guide for that, because I have a really simple table for you guys to use. Let's see, I'm going to do a human one for you guys now. Let's find a question that I have here. Let's do this one. This is the last one for today, because we've had a long session now, and I know you guys want to get back to studying and you also want to take a break. So this will be my last one. Okay. A diagram below represents the pelvic structure of and the ventral view of the skulls of 3 organisms. They are drawn to scale. So I just want to point out that when you look at this, everybody, don't read it like downwards. It doesn't mean that these 2 structures belong to each other. It doesn't mean this pelvis belongs to the skull. It just means we have a column of pelvises and we have a column of skulls, all right? But already you should be able to identify who's who in this picture. Okay. Sunita, sorry, I can see your question. Don't worry. I'm going to answer you now. I'm going to come back to you, Sunita, so I can do this one. I promise I'll go back on the blood groups. Write down the letters of the organisms that represent the skulls of bipedal organisms and the pelvic structure of a quadruped. So first things first, the skulls. Let's have a look at those. The clue of who is bipedal is this structure over here, the foramen magnum. And we should know that the foramen magnum, when it's more forward, is bipedal. So out of these three, who is more forward? Well, it's definitely Z and it's definitely X. They're very, very similar, but Y is definitely a no-no. And then the pelvic structure of a quadruped. So if we look at the pelvises, these two pelvises look very similar to one another. This one is the one that we need to look at because what it is, isn't it very narrow and long? And those are the two things that quadrupeds have. So organisms that walk on four legs. So that's going to be organism C. Okay. Then it says, give a reason for your answer in 315B. So this one, well, my answer was the pelvis is narrow and long, and that's what will give you 2 marks. Okay. And last but not least, describe one other structural difference between a bipedal and a quadrupedal organism for 3 marks. So they mean one other difference than what is in these pictures. And it has to be a structural difference. So you can't pick something random like the canines. Okay, you have to pick something that is linked to being bipedal and quadrupedal. And the main thing, the difference, is the spine shape. So you'll see it's for 3 marks. So what you're going to have to do is describe one other structural difference. So I'm going to say here, for my answer, I'm going to say bipedal is S-shaped. Quadruped is C-shaped. And the shape of spine is linked to movement. And that gets me my 3 out of 3. 1, 2, 3. That's it. Okay, I'm going to go back to Singita's question because I just want to answer her quickly about blood groups. So Singita's question is, if parents are A and B, how is an O child present, or is an O? Okay, great question. So if I have a parent that is A and I have a parent that is B, and somehow they produce a child that is O, right? Is this possible, or is this child adopted? Well, the answer is yes, this child is possible. Because if we look at our options above, if the mother has these alleles and the father has these alleles, you'll see they both have a small lowercase letter. So I'll write them down here. So if the mother is this and the father is this, to make this child inherited a lowercase I from mom and a lowercase I from dad. And therefore, this is possible. Okay? Let me tell you what's not possible. Maybe that will also help. Let me give you a different situation, and maybe that'll answer your question. If I had the following, if I had A, B, and A, can I have an O child? Well, let's do the calculations. This parent is these alleles. And this parent is A, so she could be this or this. Can you have an O child? Well, an O child must be 2 lowercase i's. Now, they could potentially get the one from there, but can they get it from the other parent? No. So this child must be adopted. Okay, this is not their child. You can use this for paternity as well. It's not a great way to determine paternity, but if this is the dad and this is the mom, this can't be his child because he doesn't have any alleles that is shared with this child. I'll do one more for you before I go. Let's do another version of this. Let's say we had B, parent, and we had AB, and the child is A, right? is this possible, or is this child adopted? So if I fill in their alleles, this parent is AB, like that. This parent is either going to be that or this. And now this child is A. So our options for A are going to be this or this. Now, are these possible? The answer is yes. Let's have a look. Okay. So this parent over here gave this individual their A letter. Okay. So there it is on either one. But now to get the other letter, if we go look at our B parent, which of these two options must this parent be to get this? Well, if this parent was these two big B's, we wouldn't be able to have this child. The only way to have this child is with this option here, because then this small I is given to the child here, and now we have the correct alleles. So this is possible. So this would be the combination that we would have to make this child. Okay, so that's how you kind of calculate how babies are or are not related. Okay. I think that's it for today. We had a very long session today. I am going to upload this onto my video, into my video playlist. If you want to come back and watch something, I'm going to put the chapters in so you can fast forward to different sections as well. And that's it for today, everybody. Good luck for Monday, and I will see and speak to you all soon. Bye.